Hello, I'm Dennis Gross from Texas A&M University in the Department of Plant Pathology and Microbiology. And I'm a bacteriologist who is studying plant pathogenic bacteria, and I'm working on Librevector and trying to develop some diagnostic procedures that will be improved over existing for more rapid and quicker and more specific in the field. So we're taking a couple of different approaches right now. The first one is to take the conventional PCR approach, and in this past year I think we've made a lot of progress in identifying some primers that amplify a region that seems to lead to better detection in the samples as compared to existing primers. And these then are being used in collaboration with Dr. Betsy Pearson in horticulture department at Texas A&M in looking at the movement of the bacterium in the phloem of the infected potato tissues. The other approach that we're taking, which we're really quite excited about, is called LAMP for short. It's an isothermic loop-mediated amplification PCR procedure that doesn't require a lot of expensive PCR equipment, and it's just very rapid. You can do this in the field potentially within an hour. It's a procedure that's been used in the medical field, and I think it shows a lot of promise to be just a lot faster for us to be able to get some diagnosis there right in the field. So we still have a ways to go yet in defining the primers and conditions that we want to use, but we think it's going to be the future for PCR detection in potatoes. So what we hope to do also is to look at the different populations of the pathogen with some of these new primers and detection methods, and get a better feel if there are different kinds of or different populations of the Librevector in South Texas or in North Texas or Nebraska. So we'll begin to look at the population structure. We don't believe that they're all going to be the same, but we're going to expect to see some diversity in the pathogen. But at this point, we're just finally getting to the point where we can actually address this issue and look to see if we can actually track the pathogen here in Texas. And this again will facilitate in our ability to control this pathogen in potato. So we look forward to the future, and we're happy to be working with AgriLife Research and Texas A&M University in conducting this research on potatoes.